recording. And yeah, so let's keep talking about the fluid mosaic model that we ended up with on Monday. So we were just chatting about how, according to the fluid mosaic model, proteins that are involved with the membrane, as long as they're not constrained by anything, are able to undergo lateral diffusion freely, as well as any of the lipids that are in the membrane. They are, for the most part, able to diffuse laterally pretty freely unless they are attached to something or constrained by something. And so that brought us to the second part of the question, which was, what does FRAP stand for, and how is it used to observe lipid and protein diffusion? So I'll give everybody, I know we ended off with like thinking about this last time, but I'll give everybody like 2 minutes to look over what they said on Monday and remember what they were thinking about before we left on Monday, and then we'll get back into it. All right, so hopefully that's enough time to refresh ourselves. So first of all, whoa, what just happened? Interesting. Okay, what does FRAP stand for? Anybody? Yeah. Yeah, exactly. So FRAP stands for fluorescence, recovery, after photobleaching. So FRAP. Hopefully you can all read that. So how is it used to observe lipid and protein diffusion? Or like what are the steps you have to do? Yeah. Yeah, so that is a good example that demonstrates mobility of proteins in the membrane using fluorescence, but that is not FRAP. So FRAP is a different technique. Yeah, do you want to go ahead? Yeah, exactly. So the key with FRAP, so like your description of that experiment was a very good experiment, but with FRAP the key word is photo bleaching, right? So we're going to use a laser to photo bleach. So but before we do that, we have to first fluorescently label that the proteins or the lipids that we're studying. So either of these experiments start this way, right? You have to fluorescently label what you're interested in observing, the movement of. And then once you've done that, like Jaheen said, we start to photo bleach a really small area using a laser. And when we photo bleach using a laser, for something that's fluorescent, once it's been photo bleached, it cannot be fluorescent again. It's done. So, like if you have this small area that's photobleached, if the lipids and the proteins on the surface of your cell were not able to laterally diffuse, you would always just see a dark spot there where you had the photobleaching, where you're not seeing fluorescence. But if you leave the cell for time and you wait, and the lipids or proteins that you labeled are mobile, they're able to then diffuse laterally into that spot that was photobleached. So it looks like over time there's recovery of fluorescence in that small area. And it's not that the lipids that were there regain their fluorescence. It's that other lipids or proteins that were fluorescent moved into that area. So those are essentially the 4 steps. You label what you want to study, you damage one area with a laser, you wait, and then you look and see if anything moved into that area that you damaged. And so why is this a useful technique? What can it tell us? Yeah, exactly. So it can tell you how fast particular proteins or lipids are diffusing. And it, and you can also use it to measure different things that are on the cell surface. Maybe you'll label 2 different proteins, 2 different colors, and then you can see maybe one moves faster, one moves slower. You can compare. If 2 things are moving at the same rate, maybe they're attached together, or if you can, but yeah, those are all the sort of types of things you can look at with this type of experiment. So you can see if the components that are on the membrane surface are mobile or if they're constrained by something. And it can tell you how fast they go, if they have any traveling partners, that kind of thing. Any questions about FRAP or the fluid mosaic model? All right, so let's start talking more about lipid synthesis and trafficking. So take a couple of minutes and look at these questions. So different steps in lipid synthesis and trafficking, and tell me where they're happening. So we'll go over that together in a couple minutes. Okay, so number 1, where are lipids synthesized? Yeah, the smoothie are. And where are they processed? Yeah. Yeah, the Golgi. And then after they've been synthesized and processed, how are they transported between organelles and to their target membranes? Yeah, vesicles. Yeah. So, and we talked a little bit last time about how vesicles are very good because they're just little tiny bilayer membranes, right? They're really good at encapsulating hydrophilic things in the center. Where would lipids go in a vesicle when they're getting transported? Yeah, exactly. So the lipids are incorporated into the membrane bilayer as part of the vesicle. All right. Any questions about lipid synthesis and trafficking? Yeah. I mean, there's a variety of vesicles. So I guess if you're specifically talking about secretory vesicles, that usually means vesicles whose destination is the plasma membrane. But you can, I mean, all the organelles in a eukaryotic cell are membrane bound, right? So eventually you'll need to make lipids to send and replenish all of those organelles as they're. I mean, one way you can sort of think about this is your cell is constantly sending vesicles all around from the ER to the Golgi, back to the ER, out to the plasma membrane, out to various organelles. And every time you're pinching off a vesicle, you're taking away some of the lipid content. Every time a vesicle merges with another membrane, it's adding on to some of the lipid content there. And like one of the cell's jobs is to stay the same size, basically, right? You don't want it to shrink if it's making too many vesicles. You don't want it to grow if it's sending too many vesicles out to the plasma membrane. And so there's a variety of vesicles kind of going all around the cell, delivering information and lipids and cargo to various parts of the cell, but also helping the cell to maintain homeostasis. Yeah, there's some really interesting things with that. Like there's different cholesterol content in different types of membranes throughout your cell, in like some organelles versus in the plasma membrane. And there's different lipid content in various organelles. And so your cell does a lot of work to maintain all of those particular lipid content in various membranes so that each organelle can carry out its specific function. But that is beyond the scope of this class. So let's, unless anybody has more questions about that, we will move on from lipid synthesis and trafficking to similar but different. We have protein synthesis and trafficking. And so here we're talking specifically about membrane and secreted proteins. And so everybody take a couple minutes again and think about protein synthesis and trafficking for these 4 questions. All right, so let's start going over these questions about protein synthesis and trafficking. So number one, membrane and secreted proteins are synthesized where or by what? Yeah. Yes, in the rough ER. And there's actually a second answer choice that is also correct for this one. Yeah. Yeah, by ribosomes, exactly. So ribosomes are very important for all protein synthesis. And for membrane and secreted proteins, ribosomes translate proteins directly into the rough ER. So at that point, number two, these proteins undergo co-translational modification where? Yeah, still in the rough ER, exactly. So during the process of translation, they're in the rough ER. So if we're talking about a co-translational modification that's happening while the protein is still being translated, has to be in the rough ER. Protein hasn't gone anywhere yet. Because the ribosomes themselves are not doing the post-translational modifications. It's usually, depending what modifications you're doing, it's a variety of other enzymes that are internal to the rough ER, like inside the lumen of the rough ER. The ribosome itself is still on the cytoplasmic surface of the rough ER. The ribosome never goes inside, but there are essentially little pores on the outside of the rough ER that the ribosome kind of sticks the end of the protein into, and then it kind of translates it like through that little space into the inside. All right, so number three. So where do post-translational modifications happen? Yeah, the Golgi, yeah. That's number E. And just to briefly think about our sugar stuff from last week, which one of these is where we're going to see N-glycosylation starting? Yeah, the rough ER. Yeah, and like this is an example of co-translational modification. And what about O-glycosylation? Yeah. Yeah. So those are two examples of co-translational modifications and post-translational modifications that both start in very specific regions. Yeah. Yeah. So from my understanding of it, most or all of the folding is done by the time the protein's leaving the ER. Most of the folding chaperones are located in the ER. The Golgi is usually a place for editing. So it's really for post-translational modifications that are happening once the protein is folded mostly into its final shape. It might do things like add a disulfide bond or attach different functional groups, or sugars or whatever else. But generally, by the time the protein gets to the Golgi, you can consider it mostly, if not completely, folded. Yeah. All right, so that brings us to number 4. So these proteins are transported between organelles and to their target destinations in which one of these? Yeah, in vesicles, yeah. And so I'll ask you the same question I asked about the lipids. So vesicles are little tiny membrane bilayers that have a hydrophilic inside and hydrophobic membrane area. So if we have a protein that is an integral membrane protein, where would that be in the vesicle? exactly. So it'll be part of the lipid bilayer. So when the vesicle is budded off from the rough ER, part of that budding will contain the integral membrane proteins that are coming along with that vesicle. On the other hand, if you have a protein that's going to be secreted, sent to the outside of the cell, where would that be in a vesicle? Yeah. Yeah, exactly. That would be part of the hydrophilic inside. The secreted protein hangs out in there as cargo, and then once the vesicle merges with the plasma membrane, that will essentially open it up and let the secreted protein leave to the environment. All right, any more questions about sort of overview of protein synthesis and trafficking? Cool. So a little bit more about vesicles. So now we're going to talk more about proteins. So everybody take a couple of minutes and think about which proteins are helping with formation, transport, and or fusion of vesicles at these various locations or in between these various locations. All right, so who wants to start us off with clathrin? Where do we see clathrin being used for transport? Yeah. 3, yes. So clathrin is absolutely used to help make vesicles that go from the trans-Golgi to the plasma membrane. Where else might we see clathrin being used to help form vesicles? Yeah. Yes, yeah. So clathrin is commonly used for endocytosis, and that is what 5 is showing us. So the plasma membrane taking in contents from the outside of the cell. That is occasionally done, or often done, with the help of clathrin to help get that vesicle started. Where else would we see clathrin? Kind of similar to 5. Yeah, 6, yeah. So vesicles going between the plasma membrane back to the trans-Golgi also tend to have a clathrin coat. So those are the main places where you see clathrin, helping to form vesicles. And so, so now let's do COP1. So where do we see COP1 coats? Yeah, 2, yes. So moving between different areas of the Golgi network. So from the cis Golgi to the trans Golgi. Where else? Yeah, 7. Yeah, exactly. From the trans Golgi back to the cis Golgi. Do we see it anywhere else? Yeah. 8, exactly, yeah. So any vesicles that have to go back from the cis Golgi to the rough ER also uses COP1. Cool. So that brings us to COP2. Where do we see COP2 coat proteins? Yeah, 1, exactly. So COP2 proteins are helping to form vesicles that are going from the rough ER to the cis Golgi. And that brings us to snares. When are we going to see snares being used? Yeah, 4. Yeah, essentially, it's I borrowed this diagram from Dr. Middleton, and I personally struggle with like the difference between 3 and 4. There, for me, like 4 is almost like the formation of exosomes, which I don't think involves this process at all. But certainly when you're going from the trans Golgi to the plasma membrane, right, which would be more 3 in my mind, that would be definitely where you're using snares. If you're going from the plasma membrane and forming a vesicle that buds out, I don't think you'd use any of these things, to be honest. But yeah, when you're using coat proteins, they essentially help coat what becomes the outside of the vesicle, which in this case would be outside of the cell. So I would have to read more about that, to be honest. Yeah. Right. So they're involved in different parts of the vesicle process. So clathrin is really important for helping to start off the vesicle. So like if you imagine your plasma membrane looks mostly flat, and to form a vesicle out of it, you essentially have to like pinch off a piece of it. And making that little pinchy area is very unfavorable, right? Like the bilayer doesn't really want to bend that way. When it's that curved, you start inviting like water into all those little gaps. And so these coat proteins like clathrin will essentially like coat this little part, or really they'll start here, and then they'll essentially induce forces that help to make the vesicle start to form, and eventually there are other proteins that help them pinch off. So the clathrin is really helping you make a vesicle out of a flat, or relatively flat lipid bilayer. Snares are doing the opposite. Snares are when the vesicle arrives at its destination to help the vesicle fuse with the destination membrane. So snares help bring the vesicle, so like we have our vesicle now. So snares are going to help the vesicle come really close and approach this membrane by essentially like twisting together, right, to help them get closer and closer until they eventually merge. So one is like the beginning of making a vesicle, one is the end of making a vesicle. Or getting rid of the vesicle, I guess. Yeah. Any questions about that? Cool. So let's think a little bit more about snare complexes. So everybody take a few minutes and think about why a drug that would interfere with the disassembly of a snare complex would block neurotransmission. So does anybody want to get us started on why this would happen? Yeah. Yeah, basically. So we were just talking about how snare complexes help vesicles merge with their target membrane. And so this is important for neurotransmission because in neurons we have vesicles that are filled with neurotransmitters that are just waiting there on the end of the cell. When they get a signal, they'll merge, and then the neurotransmitter is able to exit that neuron before the synapse, right? So here's a synapse. So yeah, and so the way that we would normally see this happen is we have snares, right? We have the R snares that are on the vesicles, we have the Q snares that are part of the membrane, and eventually, as the vesicle approaches the cell membrane, they're able to merge together. And then we have a problem, right? So if we block the disassembly of a snare complex, now we might have released some neurotransmitter, right? But we have the snares are all kind of stuck here, right? So we have before, we have after. So not the most beautiful picture, but you get the idea, right? So now we have all these snares that are kind of all tangled together and tied up. If you want, and you know, there's still going to be more vesicles here that need to get released, right? Just one vesicle is not going to help you. And so you would eventually, maybe all these vesicles have the R snares on them, but eventually, if all of the Q snares get tied up and they're unable to disassemble from the previous vesicle, right, that they merged with, you'd run out, right? Eventually, there just aren't enough free snares on the surface to help merge new vesicles, right? So ultimately, this would stop neurotransmission because the vesicles are going to need snares on the target membrane to actually merge and release the neurotransmitter. So yeah, you can't reuse those Q snares that are sitting here, and then nothing happens anymore. I don't know. Does that make sense to everyone? Does anyone have questions about how that works or how that might work? Yeah. Yeah, sorry. I'm gonna use more colors. Let's see if that helps. So we have, I'll make a better after picture. How about that? Okay, so when we got started, we had our vesicles, they have green snares, right? The target membrane has these red snares, let's say. So if you have this one vesicle merging with this one target membrane, fine. But then after the vesicle merged with the target membrane, then I guess there are also lots of other vesicles here, right? They all have snares. They all want to merge with the membrane. But once so after the snares help the vesicle leave, they're all twisted together, right? The green one from the vesicle and the red one from the original membrane. And they have to be able to essentially unentangle from each other. They have to disassemble so that the green ones can go onto a new vesicle and the red ones can stay there in the membrane and wait to help the next vesicle merge. If you block this disassembly process, eventually you run out of red ones, right? They're all tied up with green ones, and then you can't have any more release of neurotransmitters from that cell. Does that make more sense? Okay. Yeah. Okay. So, any more questions about that? Cool. All right. So, we're going to take a little interlude now with our favorite virus, SARS-CoV-2. And so now we're going to talk more about integral membrane proteins. So, so let's start talking about our spike protein. So as we know, the spike protein is on the surface of this virus. It's a transmembrane protein, right? It has this transmembrane domain that's down here. It's a trimer. So in this picture on the left, you can see like these three different colors, the pink, the yellow, and the green. Kind of shows up in the projector. It's a trimer. And they're all all the pieces are kind of mushed together, for lack of a better term. And when they're in this pre-fusion, when the spike protein is in this pre-fusion state, it kind of looks a little bit like you see in this first part of this diagram. And eventually the spike protein is going to find its target receptor, right, which is the ACE2 receptor, which is shown up here at the top in this orangey color. So how does the spike protein actually interact with the ACE2 receptor and get itself inside of your cells is the question. A few things have to happen first. So the first thing that happens, or an important thing that happens, is the transition to this state where this is like the open spike state where it starts to interact with the ACE2 receptor. And the way this happens is through this really important process that is being shown here with this furin protein. So furin is a protease. It helps to cleave peptide bonds in between amino acids. And if you look at this diagram on the left again with our spike protein in general, it has these sites, these S1, S2 sites, and this S2 prime site. So the spike protein, before the virus is able to get into one of our cells, it actually has to split into two pieces. And it does this by splitting from just one big spike protein into S1 and S2. And the way this happens is one of the proteins, as part of our body, a furin protein, which is a protease, is going to cut at this site in between what is going to become the S1 and the S2 pieces. And it has to do this 3 times, one on each of the trimers, or one on each monomer of the trimer, right? And then when this happens, this is what allows for essentially a change in conformation that allows this S1 piece to start interacting with the ACE2. The S2 piece kind of separates, and then ultimately the S2 is going to do this wild conformational change, and so it becomes really elongated. Now it has a lot more alpha helix character, and it essentially is able to then penetrate the membrane of the host cell. The S1 piece of the protein remains attached to the ACE2 receptor. It's kind of the ACE2 receptor kind of starts off the process, but it's the S2 piece of the original spike protein that will actually help to penetrate the host membrane. And then ultimately, there's a lot of other changes that happen that will start going into that allows the membranes to fuse. So, so this part was really more about the proteins. In this chapter, we're thinking more about lipids and membranes, so we'll sort of transition over to that. So here is a more wide overview of what this process looks like. So there's 2 different models for how the SARS-CoV-2 virus will enter one of your cells. So one is called endosomal entry, and one is called cell surface entry. So for endosomal entry, that looks a lot like what we were just describing, right? You have this virus, a piece of it interacts with this ACE2 receptor on the cell surface. Ultimately, and this is not a very accurate picture that shows the cleavage and the S2 protein actually doing this whole process. But ultimately, right, the cell can undergo endocytosis, and it'll pull the whole virus in as it's attached to the ACE2 receptor. It'll put the virus into an endosome. In that endosome, there's a second cleavage event. So that S2 protein now gets cleaved. And when it's in the endosome, a protein called cathepsin L does this cleavage. And this is ultimately going to help to merge the viral membrane with the membrane of that endosome, which you're seeing here, which spills out the the virus guts, right? The virus's contents into the cytoplasm of the cell. Similar but different, we have a cell surface entry. So this type of cell entry is also going to involve two cleavage events, but they're a little bit different. So our initial virus binding is the same. We have our spike protein binding to the ACE2 receptor. But instead of an endosome starting to form to pull the whole viral particle inside the cell, there's actually a membrane fusion event that happens right on the cell surface. And this happens because of another cell surface protease called TMPRSS2, which is shown in yellow here. And TMPRSS2 can also do that second cleavage of that S2 protein. So once you have that second cleavage of the S2 protein, that basically is what signals, again, the virus to release its contents. And it can do this directly from the cell surface into the cytoplasm of the cell. And ultimately, regardless of which method the virus particle picked, once the viral RNA is uncoated, it's able to then get translated by your own ribosomes and go through its whole life cycle, which we're not going to go into right now. But this little picture, this inset in the bottom, is also another sort of nice picture that shows you that elongation of the spike protein onto these like kind of long helical shapes that are kind of twisty. So like this is kind of supposed to resemble this. Yeah, so those are the, that's like a big overall picture. But let's talk a little bit more about some of those details now, and in particular, some of the conformational changes that we see in those proteins. So this set of diagrams is from a different paper, so the pictures look different. So first I'm just going to show you the pictures and how they look a little different. So we have our SARS-CoV-2 virus with our spike proteins. This picture is nice because it really shows how it's a trimer with three pretty much identical pieces. And then there's the two regions, the S1 region and the S2 region. And this also really shows nicely how the S2 region is made of these helical areas, these helical regions, or HR, right? And then once the spike protein interacts with the receptor, this picture doesn't show that cleavage event, but it does kind of show really nicely these little helices. So if you look at how this diagram shows it, you have your spike protein trimer, you have your ACE2 receptor. Once they start to interact with each other, you get this kind of shows this conformational change. It doesn't specifically tell you about the fusion between S1 and S2, but you get the idea. And then this little helical bundle here is showing you how that kind of inserts itself right into the cell membrane and helps the virus get in. So it's kind of a different picture that is showing the same kind of process. So once you have the viral entry, now we'll talk a little bit more about the rest of that viral life cycle. So we talked about those two different ways the virus can ultimately get its RNA into the cytoplasm of the host cell, and that's what's being shown here. So the virus that's part of the genome of the virus is made of RNA, right? And it's in, it's single-stranded, right? And so it's single-stranded. It goes from the 5' to the 3' direction, which is what's kind of shown here. And the first step that happens generally after that is your cells don't directly just start translating like that original RNA genome. The first thing that you often will do is make a special polymerase that actually helps you copy that RNA to make a template strand that is the complementary strand to the genomic RNA. And then you can make lots and lots of these template strands using the special RNA polymerase, which will ultimately let you have lots and lots of templates that you can use to translate and make lots of protein at the same time. So does anybody, I think I've mentioned this before, does anybody remember the name or the type of special RNA polymerase that the virus is going to use to copy the RNA? No, but RNA polymerase, you're on the right track. So, all right, so we learned about RNA polymerase a while ago. So unusual, in general, the RNA polymerases that we are using, they're using DNA as a template, and they're making an RNA complementary strand, right? That's how our cells those are the types of RNA polymerases that we have in our cells. We're using a DNA template, we're making RNA. Is that what's happening for SARS-CoV-2? What is the template here? Yeah, it's RNA, exactly. So that's why we need a special, or the virus needs a special RNA polymerase that uses RNA as a template. Sorry? So reverse transcriptase is also a little bit different. So reverse transcriptase does use RNA as a template, but the product that it makes is DNA. So reverse meaning it goes backwards from RNA to DNA. So it does use an RNA template, but the product is DNA. So that's also not what our virus wants to use. So it's a really straightforward name. So it's just called RNA-dependent RNA polymerase, which we sometimes just write as RDRP. And so this means RNA polymerase, meaning it makes RNA, and it's RNA dependent because it uses RNA as its template. So at that point, once the virus has conveniently used our cells to help it make this polymerase and then make more RNA, then we get into the sort of rest of the virus life cycle, which I'll just go through briefly because it isn't really the main focus of this class. But so we started kind of here. We have the genomic RNA, and then we made that into that complementary RNA. And then now we, I guess, transcribe that that copied RNA and to make mRNAs that are actually specific codes for all the proteins that we're going to make. So, and then you can make lots and lots of different mRNAs for different proteins depending what the virus needs. And ultimately those proteins are then going to get translated using our own ribosomes and other important machinery for translating proteins. And then eventually all the proteins and all the replicated genomes eventually get assembled into little viruses, right? And then they have to leave your cell. So where they get assembled is happening just like with all of our other protein synthesis and trafficking, right? So you translate proteins. If they're integral membrane proteins, they're going to get made right inside the RefER, which is shown here. And then it's going to get trafficked in vesicles to the Golgi for any post-translational modifications. We talked last time about how there's a lot of different glycosylations on the spike protein. So we'll see those starting in the RefER as well as in the Golgi for the N and the O-length glycosylations. And ultimately, the virus actually does this really kind of interesting thing where the whole virus hides inside another vesicle. So it basically has two membranes, right? You have your own vesicle, and then inside it you have a virus that's also enclosed by a membrane. So we call these double membrane vesicles. And then once that outer vesicle goes to the plasma membrane, and it can merge with the help of snares, like we were just talking about, and then that will release the whole virus to the outside of the cell. So does anybody have questions about how that kind of works? Yeah. Yeah. So that is the genomic RNA, and it's sense. So it's the coding strand. If you were to directly translate that RNA, it would make the correct proteins just as like one very long protein. So then when you make a complement to that, you then have to use that complement to make copies again to go back to the original direction of the RNA.
So you get the genomic RNA out of the virus.
Yep. Yeah. And then you can make like lots and lots of copies of this complementary strand. Yeah. And then you can use the same RDRP to again, copy pieces of that complementary strand to make individual mRNAs, like for particular genes or particular reading frames that the virus is going to then use to translate into protein. So neither of these tends to actually get translated into protein. You end up making lots of like little baby RNAs first. Yeah. 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 And then it's sort of shown here. So you have this complement that we just made, and then you go through the same transcription process using our DRP again to make all these little mRNAs, which are shown here in this purple. So like these are all copied regions of this longer template one, if that kind of makes sense. All right. Any more questions about that? See thoughtful looks, hopefully good thoughts. So that is what I have for chapter 9. We're going to go on to chapter 10, but I'll give everybody like 5 minutes just to switch gears, and I'll meet you back here at 1:30.